All right, Dr. Becker again. We are moving on to chapter 4, part 2 of our lecture, and we're going to keep on a rolling. Please remember on the previous lecture to go back through and review the key concepts that were listed at the top of the PowerPoint slides. Those are a really good point to review after you're done listening to that lecture. Again, we have some of the concepts in cell signaling here. And so let's start off with a good review from our friends at Osmosis so that we can get a refresher, and then we'll talk about the book content.
To make a multicellular organism, cells must be able to communicate with one another, and to do it, cells often send out tiny chemical signals that act on the receptors of other cells. Signals can be classified according to the distance between the signaling cell and the target cell. Autocrine signals are produced by a cell and go to its own receptors, so the cell sends a signal to itself. Paracrine signals are produced by a cell and go to target cells that are nearby, and endocrine signals are produced by a cell and go to target cells that are further away. Examples of these include hormones that are secreted into the bloodstream, as well as cytokines that can be released at the site of injury and act on the brain to cause a fever. Signaling molecules, or ligands, can be hydrophobic, meaning that they tend to repel water, or hydrophilic, meaning that they tend to stay in water. Hydrophobic signaling molecules can't freely float in the extracellular space, so they're brought into the target cells by carrier proteins. Hydrophobic molecules can diffuse across the cell membrane and bind to receptor proteins inside the target cell, either in the cytoplasm or in the nucleus. Most signal molecules are hydrophilic, so they can freely float in the extracellular space to reach the target cells, but are then unable to cross the cell membrane. So to pass on the signal, hydrophilic molecules bind in receptors on the cell surface. These receptors are transmembrane proteins with an extracellular end that binds to the ligand, and an intracellular end that triggers a signaling pathway inside the cell. We can think of the cell signaling pathway in 3 stages. The first stage is reception, which is when the target cell's receptors bind to a ligand. It's like a key fitting into a lock. Then there's transduction, which means that the receptor protein changes in some way, and that activates intracellular molecules, the second messengers. The third stage is the cell's response to the signal. Zooming into these transmembrane receptors, there are 3 major classes: G-protein coupled receptors, enzyme coupled receptors, and ion channel receptors. G-protein coupled receptors are 7-pass transmembrane receptors. These are like really long proteins that have one end that sits outside the cell and binds the ligand. Then the snake-like protein dips in and out of the cell membrane 7 times, and finally ends on the inside of the cell. The end of the G-protein coupled receptor that's within the cell activates intracellular proteins called guanine nucleotide binding proteins, or G-proteins. G-proteins are made up of 3 subunits called alpha, beta, and gamma, sort of like a flower with 3 petals. The alpha and the gamma subunits are anchored to the cell membrane and keep the G-protein right next to the receptor. G-proteins bind to guanosine diphosphate, or GDP, when they're inactive. When the alpha subunit is bound to GDP, the 3 subunits stay together so the flower is closed. But when the ligand binds, the G-protein coupled receptor changes its shape, and this allows the G-protein to release GDP and bind GTP instead, activating the protein. When the alpha subunit is bound to GTP, the alpha subunit separates from the beta and gamma subunits, like one petal opening and separating from the others. When that happens, the alpha subunit is free to interact with other proteins. It stimulates some while inhibiting others. But to act on other proteins, the alpha subunit turns GTP into GDP, and when that happens, the 3 subunits come together again, the flower closes, and the G-protein is turned off. Overall, there are 3 types of G-proteins: GQ, GI, and GS, and each one stimulates and inhibits a different set of enzymes and molecular pathways. The GQ protein activates the enzyme phospholipase C, which is found in the cell membrane. Phospholipase C then cleaves a phospholipid called phosphatidylinositol 4,5-bisphosphate into inositol trisphosphate and diacylglycerol. Inositol trisphosphate is soluble and diffuses freely through the cytoplasm and into the endoplasmic reticulum, where it opens up calcium channels. Since the calcium concentration is higher in the endoplasmic reticulum than in the cytoplasm, calcium flows out of the endoplasmic reticulum to the cytoplasm. The increased calcium concentration in the cytoplasm changes the electrical charge of the cell and can lead to depolarization. Meanwhile, diacylglycerol remains attached to the cell membrane and binds to the enzyme protein kinase C, which also relies on calcium to fully activate. Once calcium levels in the cell go up, protein kinase C starts to activate proteins by adding phosphoryl groups to them. Next is protein GS, which stimulates the enzyme adenylate cyclase. Activated adenylate cyclase takes adenosine triphosphate, or ATP, and removes 2 phosphate molecules, transforming it into cyclic adenosine monophosphate, or CAMP. CAMP moves throughout the cytoplasm and binds to the enzyme protein kinase A. Protein kinase A has 2 parts, a regulatory subunit and a catalytic subunit, and CAMP specifically binds the regulatory subunit of protein kinase A. When CAMP binds, it makes the regulatory subunit dissociate from the catalytic subunit. It's kind of like pulling the pin out of a fire extinguisher, allowing it, in this case it being the catalytic subunit, to do its job. So, after dissociating, the catalytic subunit of protein kinase A is free to phosphorylate target proteins that trigger a cellular response. Finally, there's the protein GI, which is also bound to adenylyl cyclase, but in this case, it inhibits it, causing negative feedback on protein GS. This is particularly important in helping to inactivate cells. Next are the enzyme-coupled receptors. They're usually single-pass transmembrane proteins, meaning that they have only one transmembrane segment, and their intracellular end has intrinsic enzyme activity. In other words, enzyme-coupled receptors have two parts. One domain is the receptor, and the other domain is an enzyme. Each domain has a separate function, like on a Swiss Army knife that's composed of both a knife and scissors. The enzymatic domain is usually a protein kinase that phosphorylates the receptor domain. Now, there are three main types of enzyme-coupled receptors, based on the amino acid at which the receptors get phosphorylated. The first group are the receptor tyrosine kinases. These are the most common enzyme-coupled receptors, and there are many subfamilies. Receptor tyrosine kinase are generally molecules that can't phosphorylate their own tyrosine side chains. When a ligand binds, two receptor chains come together and dimerize, and they cross-phosphorylate one another at multiple tyrosine residues. This triggers a conformational change that creates high-affinity binding sites for the second messengers, which can also be phosphorylated and activated, triggering the signaling pathway. Next are the tyrosine kinase-associated receptors, which work in nearly the same way as receptor tyrosine kinases, and their name even sounds almost the same. The key difference is that they have no intrinsic enzyme activity. Instead, they're associated with cytoplasmic tyrosine kinases. When the receptors bind their ligand, the cytoplasmic tyrosine kinases phosphorylate various target proteins to relay the signal. Finally, there are the receptor serine-threonine kinases, and they have a serine-threonine kinase domain on their intracellular end. There are two classes of these receptor serine-threonine kinases, type 1 and type 2, which are structurally similar. Ligand binding brings the two together so that the type 2 receptor can phosphorylate and activate the type 1 receptor, which in turn recruits and phosphorylates various target proteins to relay the signal. Finally, there are the ion channel receptors, which are generally closed. But then they open up once they bind a specific ligand. They allow ions like chloride, calcium, sodium, and potassium to passively flow down their gradient. This leads to a shift in electric charge distribution inside the cell, triggering a cellular response. Alright, as a quick recap. Autocrine signals target the same cell. Paracrine signals target nearby cells, and endocrine signals target distant cells. Hydrophobic ligands are able to diffuse across the cell membrane and bind to receptor proteins inside the target cells. Hydrophilic ligands are unable to cross the cell membrane, so they must bind to transmembrane receptors, which have an intracellular end that triggers a signaling pathway inside the target cell. There are three major transmembrane receptor classes: G-protein coupled receptors, enzyme coupled receptors, and ion channel receptors.
I think they do an excellent job again of really just breaking that down. You can listen to that again at your leisure and read through it as fast or as slow as you would like to review that material. I'd like to point out a couple of things here. Again, we're talking about the ATP is involved as well, and how are those cells going to signal to each other. So cell membrane receptors respond to that ligand binding, and these enzymes are overwhelmingly protein kinases, which catalyze phosphorylation reactions in which a high-energy phosphate is transferred from adenosine triphosphate to the target proteins, thereby altering protein activity in accordance with body needs. This here is if we want to talk about cell-to-cell junctions. It's not outlined in this chapter and it's not testable, but if you want to listen to that to kind of help further review these foundational concepts, you're certainly welcome to. The integral membrane proteins, or the integrins, are the class of multifunctional transmembrane dimeric proteins consisting of alpha and beta subunits with an intracellular region that connects to the cytoskeleton proteins. Lots of words there. The pathophysiologic processes involving integrins include the ability of cancerous cells to proliferate without being anchored to extracellular matrix. So further down the line, when we review cancers, this comes back into play. So we would know that you should be anchored to the extracellular matrix, as it says in that first paragraph. But when you have a neoplasm or a cancerous cell, they're able to do that without it. It's an error. And so that's how those cancer cells are able to proliferate. This one here is like what they talked about in the osmosis video. The general features of a GPCR structures are noted here. These receptors are often described then as serpentine, right? Because the number of the bends in that protein as they cross the membrane are noted. Ligand binding sites for hormones or neurotransmitters face the extracellular fluid, while the receptor sites of the G protein interactions face the intracellular fluid and inner membrane. So again, they're specifically bound in a certain fashion. We get into some of the CAMP. The CAMP system is also the target of inhibitory receptors. So there's information in your book about that. And so it goes into talking about the muscarinic acetylcholine receptor. So this becomes very important to understand when we move into concepts in like neurology with Parkinson's disease and some of these medications and how do they work. It also becomes important when you're talking about this for like our heart and some of the way that our heart rates work as well. So this here is an example of the ligand binding to the stimulatory AC linked receptor, and that causes the dissociation of the attached G protein, allowing the alpha subunit S to move to and activate the enzyme AC. The beta subunits remain bound together, but can move within the membrane to modulate the activity of the other membrane proteins. So that's an example there. This here is showing the downstream effects of the CAMP, right? So as an intracellular in this, the activation of the AC by the stimulatory receptors increase the levels of the intracellular second messenger, which is the CAMP. So you can see that here. It's the inhibitory receptors like R inhibitory. On this side, they work through the inhibitory G proteins, reducing the CAMP levels down. This allows the flexible signaling. Some agonists are pushing on the gas pedal, while others are pushing on the brake. So that would be how that system works together. But then we have the downstream effects of CAMP. So as an intracellular second messenger, the CAMP binds to the inactive like pKa, and that releases the active enzyme. Active pKa then, in turn, conducts the phosphorylation of the intracellular proteins that change their shape and change their activity. So these modifications depend on addition of phosphate groups to certain serine or threonine amino acids within the target proteins. So pKa is called a serine threonine kinase. Some examples of these are shown here. Active protein kinase A and inactive protein kinase A. These are specific to the organ and cell sites where the receptor stimulation is occurring. These are just some more illustrations for us to view of how this process would work. Your book talks a lot about the GPCRs that can link a phospholipase C. So this is showing the PLC activating receptor. In this system, the G, excuse me, the G protein stimulates enzyme activity, and there are no known inhibitory receptors. So when a ligand band binds to the receptor, the G protein dissociates into alpha subunit that stimulates the PLC, and then by the beta pair that may have other physiologic actions as well. So then this happens, it binds, and you can see this dissociation here. That is going to cause the response that we're looking for. This next slide is going to show us the consequences of PLC activation, right? So it gets a little bit more complicated. The PLC activation leads to the splitting of a membrane lipid phospholipid PIP2, right here, PIP2, with the release of the DAG here, which remains in the membrane and activates then protein C, or protein kinase C, and the IP3. This then diffuses to the ER, endoplasmic reticulum, causing a burst of the calcium to be released. Most of the intracellular changes occur after PLC activation and are caused by the increase in the intracellular calcium. So this again shows us an example of how these processes work. The next illustration in your book is this one. The G proteins are GTPases with a timer function. So in this one here, let's break this down. So hydrolysis of the GTP will be by the G proteins, which limits the duration of the hormone receptor actions. Okay, so we're on a timer function. So this can be thought as of a timing switch. Okay, so in A here, no ligand bands are noted. So there's no ligands that are binding receptors, right? The receptor is inactive, and the G protein is bound to GDP. But then so this is empty, but then the hormone or the neurotransmitter will bind to the receptor. GDP is out and it's replaced by GTP, and the G protein will dissociate into alpha and beta subunits, right? So right here, they're all together. So the GDP goes out, the GTP will come in. These guys are going to dissociate here. So in this one, the subunit activates target enzymes and proteins. So the brakes are built into the gas pedal. And then when you get over into here, the alpha subunit over here, intrinsic GTPase activity splits, right? So this then splits to GTP and GDP again, binding the GDP and the beta subunits, and then we'll return it to the inactive state and kicks off that binding here. So a G protein A subunits also bind and split the GTP. So the GTPase activity starts, and we talked through that. But once the GTP has reverted to the guanosine diphosphate, the GDP, the protein then is inactivated and it requires an additional ligand binding to reactivate. So it's a cycle that'll go then, like that. Cyclic GMP is also a very important cellular messenger, intracellular messenger. And this is the enzyme guanylyl cyclase, and it's found in the retinal photoreceptor cells and in smooth muscle cells of our organs and blood vessels. So this is another process here where the GC will convert to GTP to cyclic guanosine monophosphate, which is the cyclic GMP. The cyclic GMP then acts on our intracellular targets to transduce visual stimuli or to produce vasodilation. So we will hear again about cyclic GMP when we talk about vision and cardiovascular. This is an example of a receptor with intrinsic enzyme activity. So this type of receptor has an extracellular ligand binding site, which crosses the membrane once and has an intracellular enzyme site consisting of the tyrosine kinase enzyme. Okay. So once the insulin receptor, remember, will have the two identical subunits linked by a disulfide bond, with each subunit consisting of two protein chains also linked together by the SS disulfide bond. Once two insulin molecules have been bound to the insulin receptor, the intracellular portion of the receptor becomes phosphorylated, and its own tyrosine kinase activity begins to phosphorylate nearby proteins. Okay, that's a lot of fancy words. I don't expect you guys to memorize these processes. What we're showing is that this illustrates how these work. We need to understand the general concepts because, like we've spoken of in another lecture, the tyrosine kinase inhibitors we're going to see again when we talk about cancerous cells. And those are some of the drugs that can help stop oncogenesis or initiation of cancerous transformation of cells. So you're going to hear again further down the line, both in this class and in pharmacology, tyrosine kinase inhibitors. So this is just an illustration visual to help us understand the process of what's happening. Here's another one for us to look at. So this is the tyrosine kinase linked receptors. They can be present as monomers, and they develop a full activity when a divalent ligand or two ligand bands create a dimer with intracellular phosphorylating subunits. Again, a lot of words, okay? At the end of the day, what this is going to show us here is that some of the tyrosine kinase type receptors will stimulate cell proliferation, and these are vital in wound repair and cancerous transformation of cells. We want to focus on these general concepts so we can see these things and at least kind of sort of make sense of them and understand what it is that we're looking at. The book does talk a little bit about the other well-studied receptor enzyme complexes, the EGFR, because the EGFR is present in many cancerous cells, especially those in colon and breast. So you'll see things like they're EGFR positive or negative. And these are important characteristics of these cancer cells because it helps us determine what kind of drugs that they could be responsive to. So if we can give medications that can help break up this process and these different receptors and how they bind, then that can help us get some of our different drugs that we have and help us figure out the different options we have for treatment for cancers. So continuing on that EGFR, you can see here the enzyme-linked receptors can turn on multiple pathways, right? So activation of this EGFR turns on the PLC pathway, and then these RAF pathways and the phosphoinositide 3K pathway here. And these all promote cell division and proliferation. So you will have drugs that are EGFR inhibitors as a strategy for cancer treatments. You'll see a lot of JAK. You'll have JAK positive or JAK negative components in a lot of some of the blood cancers or blood proliferation type diseases. And so this just explains that here about how these pathways work and if we make drugs that can help interrupt these things. So this one here is the Janus kinase signal transducer and activator sequence of activation, and it's strongly associated with the cytokine action in the target cells, including leukocyte proliferation and immune cell signaling. It's also something that can really be present when we have some autoimmune disorders as well. Then we have this example of the TGF-beta receptor intracellular pathway. It's very similar to the tyrosine kinase in linked receptors, but it differs because it has a serine threonine kinase activity rather than tyrosine kinase actions. More examples of these different pathways. So signaling by nuclear receptors, the hormone response elements, HREs for steroid and thyroid hormones, are often found bound to the nuclear DNA. Hormone binding initiates changes in the transcription and translation. Slower onset, but it has longer lasting mechanism of action than the cell surface receptors. So these receptors can be bound to the nuclear DNA as well. So in summary, the cells throughout the body are controlled by extracellular signals generated by the autonomic nervous and our endocrine system, as well as by immune cytokines and local damage signals. Remember that most of the signaling agents are hydrophilic. They work well in water, and they have their actions mediated by cell surface receptors, many of which are those G protein coupled receptors that increase the cell concentrations of second messenger molecules and can activate things like the serine threonine protein kinases. Okay. The ligands promoting cell division during embryonic growth and at other times of accelerated proliferation, including our immune system, they will work through enzyme-linked receptors that have a tyrosine kinase mechanism. And then we have those nuclear receptors mediate inside the nucleus attached to the DNA that mediate the cellular responses to hydrophobic steroid and thyroid hormones that alter cell transcription and translation for long-term effects. Again, we're looking for general concepts that we're understanding. We are not worried about specifically asking that you memorize any of these illustrations or those kind of things at all. Next, the book jumps into contractile cells. So we're going to change courses here. So let's start with a refresher osmosis video about contractile cells.
The human body consists of hundreds of muscles, which come in all different shapes and sizes. Each muscle's particular structure allows it to perform a specific function. The muscles are attached to bones or other tissues to help us maintain position, perform movements, and even protect some organs. Okay, now muscle tissue is made up of contractile cells, often called muscle fibers. Muscle tissue can be grouped into three types: skeletal, cardiac, and smooth muscle. Skeletal muscles connect to the skeleton and other structures like the eyes to help with movement and stability of the body. These muscles are voluntary, meaning that we have active control of them to perform movements, like flexing your elbow. Cardiac muscle is the muscle tissue that makes up the walls of the heart. These muscles contract in a rhythmic way to pump blood to the whole body, and they are involuntary, meaning that we can't consciously control this type of muscle. Lastly is smooth muscle, which mainly lies in the walls of blood vessels and hollow organs. In blood vessels, smooth muscle helps contract the vessel walls to alter their diameter, which helps control blood flow. In hollow organs, smooth muscles perform rhythmic contractions called peristaltic contractions, which moves the contents of these organs in one direction, like food in the stomach or small intestine. Smooth muscle is also under involuntary control. Alright, now muscles come in a variety of shapes that help serve their specific functions. For example, a flat muscle has parallel fibers and often has a flat, sheet-like tendon called an aponeurosis, as is the case for the external oblique muscle covering the abdomen. Next is a quadrate muscle, which describes a square muscle with 4 equal sides. An example of a quadrate muscle is the famous six-pack, anatomically called the rectus abdominis, which is a long, paired muscle that is divided into square-like portions by bands of connective tissue. Pennate muscles, on the other hand, have their fibers attaching obliquely to a tendon. These muscles can be grouped into unipennate, bipennate, or multipennate muscles depending on the relationship between the muscle fascicles and the tendon. Unipennate muscle fibers go in one direction and merge on one side of its tendon, like the extensor digitorum longus muscle in the leg. Bipennate muscles look more like a feather, having oblique fibers on both sides of the tendon, like the rectus femoris of the anterior thigh. And multipennate muscles have fascicles in different directions, attaching to a branched central tendon like the deltoid muscle, covering the shoulder. Next are fusiform muscles, which have a thick muscle belly that becomes tapered at both ends. An example of a fusiform muscle would be the biceps brachii. Speaking of biceps brachii, multi-headed or multi-bellied muscles have more than one head of attachment or more than one contractile belly. Both the biceps brachii and triceps brachii muscles have 2 and 3 fusiform heads respectively, and thus could also be referred to as multi-headed. Examples of multi-bellied muscles include the gastrocnemius muscle in the leg or the digastric muscle under the jaw, which both have 2 bellies. Next up are the convergent muscles, which are large muscles that arise from multiple points, but their fibers converge to insert into a single point. A good example is the pectoralis major muscle of the anterior chest wall. This muscle arises from the sternum, ribs, and clavicle, but inserts into a single spot on the humerus. Lastly are circular or sphincteral muscles, which are indeed shaped like a circle. Typically, these muscles surround a body opening, and their circular shape causes constriction of the opening during contraction. For example, the orbicularis oris surrounds the mouth, and when contracted, it helps constrict the oral opening, seen when puckering your lips when whistling. Okay, now let's take a deep breath and have a quick quiz. Can you identify the shapes of these muscles?
Don't worry about this. It's a lot of extra little information here.
Now, muscles attach to different body parts, including bones, cartilage, skin, or even other muscles. For example, many facial muscles attach to the skin of the face, which allows facial muscles to move the skin of the face to produce facial expressions like.
Stop there because it gets into a lot more muscle stuff than what we need at this point in the course. Okay. Both striated and smooth muscles will achieve their contraction by cross-linking of actin and myosin fibers. And so we're going to kind of break this down and talk about how you make the muscles work. So again, it's energy potential. So we need muscle contraction, and it's going to use calcium and ATP to initiate and repeat the cross-link formation. The point here is that the regulatory mechanism differ between the two muscle types. So our striated skeletal and then our cardiac muscles cells will contract in response to actin fiber modification, whereas the smooth muscle cells contract in response to myosin modification. This here talks about the striated muscle cells. They appear to have stripes, and it breaks that down for what that means. So this is a sarcomere structure. So the microscopic appearance of the skeletal and cardiac muscles will show this striated appearance. The repeating unit is the sarcomere, the functional unit then of the muscle contraction. So the sarcomeres span from Z disc to Z disc, as seen here. Darker A bands flanked by lighter I bands and an M line at the middle of the sarcomere. I don't need you to memorize these things at all. We're talking about the concept here of a sarcomere to be comfortable and understand what that is. So this structure is responsible for hundreds of copies of each of the 2 linear proteins that reversibly bind together to create a muscle contraction. Actin is protein made up of repeated subunits joined together to form a thin strand, which is further surrounded in that spiral fashion, and it has regulatory proteins troponin and tropomyosin. Okay, we'll talk more about that in a minute. Then myosin is a protein containing 2 heavy chains and 2 pairs of light chains, and so they work a little bit different between the two. Together, these proteins are referred to as myofibrils. Okay? So myosin is the thick coiled strand with the flexible heads. So during your muscle contraction, myosin heads bind to the recognition sites on the actin strands and pull the actin strands towards the center of that sarcomere, causing the sarcomere shortening and ultimately shortening the whole muscle. Other proteins are involved, right, in holding the actin and myosin strands in alignment, and then therefore they will link the sarcomeres together and keeping those in alignment. The actin strands are held in alignment by binding to the Z discs. Myosin strands are held in alignment by binding to a large elastic protein called titin, which is also anchored on the Z discs, so which you can see here. The myosin protein powers movement as its head binds to the active sites on the actin, and then it flexes, pulling the actin filament and shortening the sarcomere, causing that contraction. In striated muscles, the actin sites are covered by the tropomyosin strands held in place by troponin. Calcium binds to the troponin and shifts the tropomyosin, and that initiates the cross-bridge formation. And then you get the relaxation occurs when the ATP, then the energy, detaches the myosin head and the calcium levels fall again. To point out here, it's just showing the heavy chains, the light chains, the regulatory chains, etc. Again, we want to talk about kind of these as global concepts. I don't expect you to memorize and be able to repeat back verbatim these things, but understanding the concepts of what we're talking about, specifically like this slide with the role of the calcium and ATP in cross-bridge formation. With calcium present, the myosin binds to the actin, the head swivels, and the filaments slide opposite each other. Then, when the ATP replaces the ADP, the cross-bridge detaches and the calcium level will drop and the cell is able to relax. So over and over again, you're having this chemical signaling and causing the triggers of the cell to contract and relax. Very complicated slides here, but essentially, we have to remember that we need ATP, we need that power, we need that energy for these muscle contractions to work. Actin filaments are tethered together at the Z zone, and the myosin filaments are tethered together at the H zone. But myosin heads are oriented to shorten the sarcomere from both ends. So between the two, the myosin and the actin, and the specific ways that they fit within the sarcomere helps regulate ultimately how our muscles contract. Remember that intracellular calcium levels are very low in comparison to the extracellular calcium. So one mechanism that the calcium can enter into the muscle cells is by action potentials that depolarize the membrane and allow them to come by diffusion. But a much bigger source of the calcium influx for contraction comes from the sarcoplasmic reticulum. So we have to understand that we have these different structures and the way that they work. The sarcolemma and the sarcoplasmic reticulum, a lot of big words here, interact to rapidly increase that intracellular concentration through a structure termed the triad, right? And then essentially you're going to get the action potential deep into the muscle fiber. You're going to get this action potential to depolarize the membrane, which signals then rapid release of the stored calcium, and that is able to cause the muscle to contract. So a lot of different steps here of how you're ultimately going to get the calcium into the cells for muscle contraction. This becomes important when we talk about things of hypo and hypercalcium, right? And helps us understand that this is, if you have too much calcium or not enough calcium, we're going to have muscle side effects for both of those conditions. Again, we talked about this term, the sarcoendoplasmic reticulum calcium ATPase circa actively transports calcium into the SR to end the contraction. These are a lot of slides, you guys, and I know these things can begin to sound just like blah, 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 and a lot of information. Again, your quizzes and your tests are open book, open notes. I just want you to have these general ideas of how these things are working. I don't expect you to memorize exactly how the sarcoplasmic reticulum stores and releases the calcium verbatim, but understand where it is in your notes and where it is in the chapter and the general concepts surrounding it. These are, again, further images that help continue to explain what we're talking about here with the triad at rest and then during activation. These are the kind of the views that we're seeing here. I like this table. I think it's very helpful. I would highlight this slide or be able to refer back to it. There's the differences between skeletal muscle and cardiac muscle, and they're outlined very nicely here. They have different forms of metabolism. They have different action potentials, and they can work very differently to be activated. We have to understand the concept then that the levels of muscle protein expression are dynamic, meaning there's lots of turnover and fluidity between how much actin and myosin, how much calcium's coming in and out of the cells, how your myofibrils are working. That we have to understand. Excuse me. So when we increase the workload of our skeletal or cardiac muscles, we will then need to increase our protein synthesis relative to protein degradation, resulting in hypertrophy. So that concept is important to understand when you're talking about strength training for skeletal muscles or hypertension control for cardiac muscle. On one hand, the decrease in the workload after an acute limb injury like a fracture can result in skeletal muscle atrophy with protein degradation at greater rate than protein synthesis. When we talk about aging, a relative increase of the muscle protein breakdown over the synthesis, so you break down faster than you synthesize, is very common in aging, and that results in sarcopenia. That's a component of frailty in older adults. Smooth muscle, loose network of actin and myosin. Remember that the smooth muscle is found in hollow organs like our GI tract or bladder or our uterus. They also make up contractile layers of the blood vessels and the sheaths. The contraction and the relaxation of the vascular and organ smooth muscle layers are regulated by our autonomic nervous system, meaning it's involuntary, but they're also regulated by local circulating chemical mediators. So smooth muscles do not have striations. They do not have sarcomeres. So if you were to be asked, what is the differences between the smooth muscle and the skeletal muscle? The answer would probably be that they have different forms. They don't have sarcomeres is a great example because they're going to work very differently. The structures are different. The smooth muscle works with actin and myosin filaments, but they're more loosely arranged within the cytoplasm, which is what we see here. So this side here in A is the relaxed, and then in B, we can see then what happens with smooth muscles are contracted. The actin myosin strands form that loose meshwork crossing the cell. The strands are anchored, remember, by attachments to the plasma membrane, which we talked about earlier, and to dense bodies within the cytoplasm. So when calcium increases, the cell shortens here. So we have that image to see here. So remember then that the action of calcium is different between skeletal and smooth muscle. Smooth muscle cells contract when intracellular calcium increases. And when that happens, it'll bind to the protein calmodulin, CAM. I can't even say that. Calmodulin. I've always called it CAM. The calcium CAM complex activates the enzyme myosin light chain kinase, MLCK. The MLCK phosphorylates the regulatory myosin light chain, allowing it to form cross bridges then with the actin. And then you get the smooth muscle contraction will continue until the calcium level falls or myosin light chain phosphatase is activated. So there are two main mechanisms producing the intracellular calcium in smooth muscles. First, there's voltage-gated calcium channels that can be opened by smooth muscle cell depolarization. Then, secondly, many smooth muscle cells have receptors linked to the GQ and signaling via the phospholipase C system. So we talked about it. A prominent consequence of this pathway is the generation of the IP3, which then causes the release of calcium from the sarcoplasmic reticulum. The example that they talk about in the book is that in the circulatory system, vasoconstriction by norepinephrine and several other circulating substances, right? So norepinephrine, angiotensin 2, vasopressin. The circulatory, we have these circulating substances because activation of the GQ-linked receptors and IP3-mediated calcium release. So the vascular smooth muscles then can help regulate blood pressure. In order to do that, they have to have the voltage-dependent calcium channels that link depolarization to calcium entry and contraction. Vasoconstrictor agonists, so they promote the vasoconstriction, so again, like norepinephrine, binds to the GQ-linked receptors, activating PLC that releases SR calcium to cause the vascular smooth muscle contraction. So all of this is happening together of the sarcolemma. The membrane depolarization of the calcium opens up and allows the calcium to come through to here. This channel also allows the calcium to come in through here when the vasoconstrictor agonist hits the receptor. So like this could be norepinephrine attaches to the receptor. This opens up this channel, more calcium floods in. We have the G-linked receptor, right? Then that is going to activate the PLC. So it's going to generate IP3, and then that releases into the sarcoplasmic reticulum, which then causes more calcium to be released, and all that together is going to cause the contraction. So again, a lot of terms and things like that, a lot of illustrations. Don't panic about it. You do not have to memorize these things. Take a breather and just make sure we understand the key concepts and the general concepts surrounding this. Okay, so when we summarize muscle cell function, we would say the muscle cells are contractile cells that will shorten upon stimulation, providing force for body movements. Our heart pumping, our muscles contracting. It's also responsible then for the contraction of the walls of blood vessels, airways, and some of our internal organs. Actin, myosin cross-linking, and cross-bridge cycling are responsible for muscle contractions, right, for our skeletal muscles. They require calcium ions and ATP for the skeletal muscles. The smooth muscle is regulated differently in various organs and blood vessels depending on the receptors present on the cell membrane. Okay, so those are very different things between our striated skeletal muscles and our smooth muscles. Then the book jumps to the next topic, which is cell injury, renewal, stress, and death. Again, osmosis does a nice job, so let's jump over to that.
Generally speaking, cells have two ways to die. One way is by apoptosis, which is a form of programmed cell death. A second way is by necrosis, which is when cells die due to injury or disease. Overall, apoptosis occurs much more often than necrosis. One example is when old skin cells undergo apoptosis and get replaced by new skin cells. Another example is in our hands and feet during fetal development. Initially, human hands and feet look like duck's feet with webs of skin connecting the fingers. But the cells in the webbing undergo apoptosis, and that allows us to form individual digits that allow us to pick our nose and play the piano. In contrast, necrosis occurs less frequently. And an example of that is when a blood vessel that goes to your big toe gets clogged, causing ischemia, which is when oxygen and nutrients can't reach the cells. As a result, those cells begin to die, turning your big toe a pretty nasty shade of black. In apoptosis, there are two activating mechanisms: the intrinsic pathway, also called the mitochondrial pathway, and the extrinsic pathway, also called the death receptor pathway. The intrinsic pathway occurs when a cell is exposed to stress, like radiation, hypoxia, or low oxygen, and high intracellular concentration of calcium ions, or oxidative stress, which is where reactive molecules with unpaired electrons, called free radicals, steal electrons from nearby molecules. These stresses cause two intracellular proteins, BAX and BAC, to move from the cytosol to the mitochondria. Once in the mitochondria, BAX and BAC pierce the outer mitochondrial membrane, making it porous and leaky. This allows two additional proteins, called SMACS, SMACS, and cytochrome C, to spill into the cytosol. SMACS binds to proteins that normally inhibit apoptosis and deactivates them. Meanwhile, cytochrome C binds to both ATP, the main form of intracellular energy, as well as an enzyme called APAF1. Together, cytochrome C and APAF1 combine to form a large protein complex called an apoptosome. The APAF1 portion of the apoptosome then cleaves an enzyme called procaspase 9 into its active form, caspase 9. Caspase 9 then goes on to activate caspase 3, and caspase 3 goes on to activate other caspases, like a chain event. Eventually, this caspase cascade leads a cell to commit apoptosis. That's because these caspases cleave the proteins that make up the cell's nucleus, organelles, and cytoskeleton, a bit like a ninja sabotaging a bridge by removing its nuts and bolts. This destroys the cytoskeleton, as well as the proteins that anchor the cytoskeleton to the cell membrane. As a result, the cell membrane starts to develop blebs, or bulges in the cell membrane. The blebs are structurally weak, so they start to break off from the cell membrane, and this attracts nearby macrophages, which begin to clean up the mess by eating up the cell fragments. So apoptosis is a neat process that conveniently recycles the organic contents of the dead cell. Now, when the signals from apoptosis come from outside the cell, that's called the extrinsic pathway. One example is when a nearby macrophage recognizes an old cell, a pathogenic cell, or a cell that's completed its task. And in these situations, a macrophage can initiate apoptosis by releasing tumor necrosis factor alpha, or TNF-alpha, which is a signaling protein that binds to an appropriately named death receptor on the target cell membrane, one example being tumor necrosis factor receptor 1. The cytosolic end of this receptor dives deep inside the cell, and it's called the death domain. When the TNF-alpha binds to the tumor necrosis factor receptor 1, the death domain changes its shape and is able to bind to two proteins. One is called fast-associated protein with death domain, or FADD, and the other is called, and it's going to take a deep breath, tumor necrosis factor receptor type 1 associated death domain protein, or TRADD. So the death receptor, FADD, and TRADD come together to form a multi-complex protein called, wait for it, the death-inducing signaling complex, or DISC. Once everything is together, DISC cleaves procaspase 8 into caspase 8, which in turn activates caspase 3, and caspase 3 goes on to activate other caspases. This initiates the caspase cascade that commits the cell to apoptosis. After that, the process of apoptosis unfolds, just like in the intrinsic pathway. Now, in addition to macrophages, if a cytotoxic T cell detects that a cell is expressing foreign antigens, the T cell will express a protein on its membrane called Fas ligand, which binds to a death receptor on the target cell called the first apoptosis signal receptor, or Fas receptor. Similar to the death domain of tumor necrosis factor receptor 1, the Fas receptor protein also has its very own death domain that can bind to FADD to form DISC. As before, DISC activates procaspase 8 into caspase 8, and that triggers the caspase cascades, which leads to apoptosis. Now let's switch gears and look at necrosis, which can be triggered by external factors like infection or extremely hot or cold temperatures, as well as internal factors like tissue ischemia. Necrosis can start in one of two ways. The first is a form called oncosis, which starts when toxins or ischemia damage the mitochondria. If the mitochondria no longer synthesize ATP, everything stops working, including the ionic pumps that regulate the flow of ions into and out of the cell. Without functioning ion pumps, sodium starts to flow into the cell, and it's followed by water, which causes the cell to swell up like a balloon. Soon, the cell bursts and spills its internal contents onto neighbouring cells, and this attracts nearby immune cells and triggers the inflammatory process. Immune cells release substances like proteases, which are enzymes that degrade proteins, and reactive oxygen species, which are unstable and damage other cells. If this inflammatory process occurs among enough cells, it can destroy the tissue, and if that happens on a massive level, it can lead to organ dysfunction. Now, it turns out that cell necrosis comes in a few different flavours. First, there's coagulative necrosis, which occurs when a tissue becomes hypoxic or has low levels of oxygen, most commonly due to ischemia. Hypoxia causes structural proteins to bend out of shape, like twisting a paperclip so that it can no longer work. Hypoxia also affects lysosomal enzymes, which become ineffective at getting rid of the affected proteins. So although the cell dies, they retain some structure and don't get completely destroyed. From a macroscopic level, the dead tissue becomes a gel-like substance and has a pale wedge shape, with the apex oriented towards the obstruction. That's because a blood vessel typically serves a region of tissue that typically fans out, and all of that tissue becomes hypoxic and then undergoes coagulative necrosis. Occasionally, blood re-enters the area of necrosed tissue, like if a blocked blood vessel opens back up, and when that happens, it gives the tissue a dark red colour, and it's called a red infarct. Coagulative necrosis can occur in any cell of the body, but it most often happens when there's low oxygen to the heart, kidneys, or spleen tissue. Next, we have liquefactive necrosis, and that occurs when hydrolytic enzymes completely digest the dead cells into a creamy substance full of dead immune cells. Think of it like a cream-filled donut. Liquefactive necrosis is most commonly seen in the brain. However, it can also happen to pancreatic cells or within an abscess located anywhere in the body. The brain has resident macrophages called microglial cells that contain hydrolytic enzymes. These enzymes completely destroy damaged brain cells, basically liquefying the dead brain tissue. Similarly, the pancreas has various enzymes like trypsin that are designed to digest food, but sometimes they get activated in chronic inflammation due to gallstones or alcohol consumption, and destroy the pancreatic tissue itself. Finally, in abscesses, neutrophils use their proteolytic enzymes to liquefy tissue, which results in pus. Next up, there's gangrenous necrosis, and it also occurs due to hypoxia, so that's why some consider it a form of coagulative necrosis. Gangrenous necrosis typically affects the lower limbs and gastrointestinal tract, and it causes the tissue to get dried up like a mummy, so it's sometimes called dry gangrene. But if dry gangrene gets infected, then liquefactive necrosis can occur, and then it's called wet gangrene. Next, there's caseous necrosis, and it's a bit of a mix between coagulative and liquefactive necrosis. Typically, it's the result of a fungal or mycobacterial infection, classically Mycobacterium tuberculosis, which causes tuberculosis. The dead cells disintegrate but are not fully digested, which leaves the tissue with a cottage cheese consistency. Next, there's fat necrosis, which most commonly occurs when there's trauma to fatty organs that have a lot of adipose cells, like the pancreas or the breasts. Trauma to the pancreas or the breast ruptures the adipose cell membranes, which makes them spill their fatty acids into the extracellular space. There, the fatty acids combine with calcium, which leads to dystrophic calcifications in the tissue that look like bits of chalk in the tissue. Now, in addition, the pancreas can undergo fat necrosis as a result of inflammation, called pancreatitis. With pancreatitis, the pancreatic cells spill lipase around the pancreas. Lipase helps digest fat, so it causes fatty acids to spill out of the fatty retroperitoneal tissue that's adjacent to the pancreas. Finally, there's fibrinoid necrosis, which is almost always found in malignant hypertension and vasculitis. With hypertension, a constant high blood pressure damages the muscular wall of the small arteries, so fibrin, a protein involved in the clotting of blood, starts to infiltrate and damage the walls of the damaged blood vessels. Similarly, in vasculitis, there's an inflammatory reaction in the blood vessel walls that causes destruction. Okay, so as a quick recap, apoptosis is a commonly occurring form of programmed cell death, whereas necrosis is a less common process where cells die due to injury or disease. Apoptosis occurs due to intrinsic and extrinsic pathways. With apoptosis, white blood cells come in and clean up the dead tissue, so it's a neat way for cells to die. Necrosis occurs because of external factors, like an infection, as well as internal factors like tissue ischemia. There are 6 types of necrosis: coagulative and gangrenous necrosis, which happen to hypoxic tissues; liquefactive necrosis, which happens because of hydrolytic enzymes; caseous necrosis, like in tuberculosis; fat necrosis, which happens when fatty acids spill outside of the adipose cells, like during trauma; and finally, fibrinoid necrosis, which is caused by fibrin deposits, like in malignant hypertension.
I don't think I can explain that any better than osmosis did, so I'm not going to try. These are just the outlines of the things that talked about in here. The increased workload of some of the cells will adapt or cause some challenges, so the cells have to adapt, and that's outlined here. That's pretty straightforward. This one, the cell response to acute ischemic injury, makes a lot of sense. Look at all these steps, though, that have to be happening before you get through this all. Loss of energy in the form of ATP initiates many pathways that result in cell recovery or cell death, depending on the duration of your hypoxia and ischemia. Cell death in acute ischemia is generally considered to be necrotic. All right, the hypothesis of aging is an important concept that I'm glad the book goes into. We are adult practitioners, so these things definitely come into play for us to talk about the characteristics of aging. Aging is not a disease process, but it's accumulation of cellular changes that may compromise function, but without necessarily shortening lifespan in an individual who is generally healthy. Age-associated changes in the organ systems are noted in each chapter, so as we go through, you'll have geriatric considerations. But if you have favorable genetics and you lead a healthy lifestyle, people that are healthy older adults can have really good organ system function for a long time. But there are some things that we see as some of the hallmarks of aging. One is cellular senescence. So cells reach a replicative limit and can no longer enter the cell cycle for production of daughter cells. I mean, at some point we all die, right? So even the most healthiest of person, we have some of these things built in that as time goes by, we are going to die. Our cells are going to stop working. We have telomere attrition. That's related to cellular senescence because shortening of the telomeres at the end of the chromosome means that the DNA can no longer be replicated. So therefore, we don't have cell division. We can have genomic instability as we age, means the failure of the DNA repair process can allow damaged DNA to accumulate. That's a problem. Then we have loss of proteostasis. So we have turnover of damaged proteins, and that is initiated by binding of the protein ubiquitin and targeting to proteasomes for degradation. So we have some things that happen as we age that come into play here. This is all under your section of the 9 hallmarks of aging. Then we have the deregulated nutrient sensing. So we have molecules will lack the proper nutrients, and these alterations can lead to deregulated sensing. Mitochondrial dysfunction has a major impact also on the aging process, because as cells age, our powerhouse, or the efficacy of the respiratory chain to the mitochondrial, tends to decrease, and we get loss of ATP. So our powerhouse powers down. We think also that stem cell exhaustion can reduce the number of cells with rapid turnover. And then we know that there's epigenetic alterations. So we have things throughout our lifespan that cause DNA methylation, post-translational modification of our histones, and chromatin remodeling. Epigenetic alterations can directly affect regulation of our telomere length. That's also we talked about earlier as a hallmark as well. Then we can talk next about the idea of frailty, and I don't know why we don't have a slide in here. Frailty is something that often is associated with aging. A risk, we consider age as a risk factor in and of itself, because the idea is that as we age, sooner or later we become to develop these hallmarks of aging, and things start to not work well. Frailty is a multi-system syndrome that marks the loss of reserve that leaves an individual at an increased risk of death, decreased in mobility, and decreased independence. We use the term frailty to indicate a multi-system physiologic changing that is occurring. You can still be classified as frail without having a life-threatening disease process. So frailty can be seen with age. You can be 103 and be very frail and not really have any disease process. And then inflammation is an important marker of frailty. So chronic levels of inflammation can be a consequence of a variety of different mechanisms. And older adults typically have some other comorbid conditions that also promote inflammation. But there's some theories of cellular aging and the role of inflammation as well. Okay, the good news is that now that we've ended chapters 2, 3, 4, we can move into much funner, if you will, body systems reviewing of physiology and pathophysiology. And we can end with all of this taking us back to undergrad, to chemistry and organic chemistry and microbiology and all of that business.